Hello, I am Abigail Rafalski, a third year at the University of Florida and research assistant for Leslie Gaynor, who is a doctoral student in the Clinical and Health Psychology Department, working under the direction and guidance of Dr. Russell Bauer. Today, I will be presenting a project titled Amyloid Positivity Impacts Trial-Specific Visual Object Discrimination Performance in Cognitively Normal Older Adults. Alzheimer's disease, also known as AD, is a progressive neurodegenerative disease that occurs in older adults at or above the age of 65. It is the sixth leading cause of death in the United States and currently inflicts more than 5 million individuals who are part of our older adult population. Efforts to diagnose AD early are integral to our effort to reduce the economic and social burden placed on older adults with AD-related dementia and their families. The presence of brain biomarkers, such as beta amyloid plaques, are an accurate pre-morbid measurement of the progression of AD. Additional factors that confer increased risk for AD include brain atrophy and a family history of dementia. However, specialized imaging techniques can be costly and can be difficult to access. Another method of detection is through cognitive testing, which offers a cheap and fast way to identify cognitive impairment stemming from dysfunction in the regions of the brain that are vulnerable to early AD pathological change. For example, visual object recognition and discrimination tests including the ORDT, have been shown to be sensitive to dysfunction in AD vulnerable brain regions, such as the anterior temporal lobe. Notably, these tests involve tasks of many types of object recognition and discrimination, including semantic and non-semantic object discrimination. In this research study, we sought to determine if the presence of AD-specific risk, such as amyloid positivity and family history of dementia, is related to performance on semantic versus non-semantic visual object discrimination trials. This project was completed by collecting data from 57 cognitively normal older adult participants from the Wine Center of Alzheimer's Disease and Memory Disorders at the Mount Sinai Medical Center in Miami, Florida. These participants completed a neuropsychological and clinical evaluation as well as an amyloid PET and MRI scans. Amyloid positivity was determined by qualitative and quantitative analyses completed by an expert neurologist. In addition to these evaluations, participants completed a computerized visual ORDT oddity task in which they were shown a horizontal array of stimuli and were asked to identify the different entity from the lineup. There were two difficulty levels, easy and difficult, with our primary focus being on difficult condition. In the difficult condition, there were 3 non-target items that were rotated to a unique plane of the same entity, which requires a participant to recognize the non-target items are the same entity from different angles, also known as viewer invariant recognition. The difficult objects had 2 trial types, animals and non-real objects. Animals category represented semantic objects, which are entities associated by recognition of their names, and non-real objects represented non-semantic entities, which are made up of random features that do not create real or recognizable objects. This data was analyzed using repeated measures of analysis of variance to determine if there was a difference between performance on these 2 trial types related to AD risk. We found that there was no significant difference in the performance of participants with a family history of dementia and participants with no family history of dementia. However, there was significant difference in performance based on amyloid positivity, such that amyloid positivity was related to worse performance on semantic compared to non-semantic trials, in comparison to amyloid negative participants. In conclusion, amyloid positivity was related to worse performance on semantic trials compared to non-semantic trials of the ORDT tasks. This suggests that semantic or animal trials may be sensitive to AD-specific risk factors, such as amyloid accumulation. Interestingly, amyloid positivity may negatively affect both semantic processing and object discrimination abilities, which are 2 cognitive capacities supported by the anterior temporal lobe. In future studies, creation of cognitive assessments that test the 2 capacities may benefit earlier detection and intervention for these older adults at risk for AD-related dementia. Thank you so much and have a wonderful day.